Good morning, everyone. We just opened the doors to the webinar, so I'm just gonna take a moment as I see the participant numbers climbing, and we will get started in just a minute or two once the curve asymptotes participants over time. I guess I can take the time as people are joining the Zoom to introduce myself. I'm Jeff Miller. I'm one of the co-directors of Grand Rounds. We're very pleased to have you all here virtually. And I think that we'll get started. So we just have one announcement before we get going, which is next week's Grand Rounds, which will be given by Dr. Stephen Paul, who is the CEO of Karuna Therapeutics. The tentative title, I think this will be the actual title of the talk, is Neurosteroids and GABA Receptors from Lab Bench to Medicine Chest. And then I'll just make a couple sort of logistical points about our session today. The first is that in response to a recommendation from an attendee, closed captioning is available throughout the Zoom. In order to access that feature, you have to be using the Zoom app itself, not joining via a web browser. And if you hover toward the bottom of the Zoom window, you'll see a live transcript button. And if you clicked that, that will turn on the closed captioning. You can also adjust the font size. So if you go into settings and accessibility, you can adjust the font size of the closed captioning. You should feel free to pose questions throughout today's talk. We will get to them during the discussion period at 12 o'clock, but you don't need to wait until the end of the talk to ask a question. If it occurs to you, go ahead and pose it in the Q&A section. Again, if you hover to the bottom, you'll see a Q&A button. You click in there and can ask a question. Please do not put the questions in the chat. That will allow us to have them all in one place so that we can make sure that we see your questions and get to them. And as a reminder, we will be prioritizing trainee questions. So if you're a trainee, please put trainee at the beginning of your question, and we will prioritize those at the beginning of the discussion. So on to today's speaker. We're really delighted to have Dr. Mary Phillips join us today. Joining us from the University of Pittsburgh virtually. And Dr. Phillips has been through a busy week of important talks. She gave a big webinar yesterday to BBRF, formerly called NARSAT. So we're really lucky that she has found time to speak with us today. Dr. Phillips is the Pittsburgh Foundation-Emmerling Endowed Chair in Psychotic Disorders and is a full professor in psychiatry and clinical and translational science at the University of Pittsburgh. She completed her undergraduate training and the US equivalent of an MD PhD at Cambridge University. And her research employs multimodal imaging, novel computational and translational approaches to identify neural biomarkers that reflect those processes to answer questions about diagnosis, early identification, and treatment of mood and anxiety disorders in youth and adults, focusing on mood and anxiety disorders. She's identified abnormalities in reward circuitry that are specific to bipolar disorder to aid in more accurate and rapid diagnosis, and to identify youth who are at risk, future risk of the disorder. And she's now determining how neural biomarkers can be focally targeted by a new neuromodulation technique, theta burst stimulation, to develop more effective treatments for bipolar and related disorders. Her CV is very impressive. A couple points that I would highlight is that she's successfully mentored over 100 junior colleagues, including 13 NIH-K awardees. And she's had presidential or council roles in major international neuroscience societies, including being past president of the Society of Biological Psychiatry. And on the topic of biological psychiatry, that organization and its journal, Biological Psychiatry, I'll note that she has 2 articles that are among the 10 most cited articles of all time in that journal, both on the neural basis of emotion regulation and perception. And then a final thing before I hand the microphone over is just looking at Dr. Phillips's current support. It's really remarkable the breadth of her interest. She has currently funded grant support in studies of bipolar disorder, anhedonia, OCD, emotion regulation, and transdiagnostic mechanisms of psychopathology. And that's just a subset of her current work. So I'm really thrilled to welcome Dr. Mary Phillips. The title of her talk today is Reward and Emotion Regulation, Using Neuroscience to Develop New Brain-Based Treatments for Bipolar Disorder. Welcome, Dr. Phillips.
Well, thank you very much. And hopefully you can all see my slides, I hope. So first of all, thank you so much for inviting me to give a talk here today. Even though it's virtually, I'm honored to be part of your program. I wish I could be in New York City in person, but this is, I suppose, second best. So as Jeff said, basically what I want to do today in the short time I have is to go through some of the studies that my lab and I have been performing over the last couple of years or so, basically trying to, in the big picture here, is trying to use our understanding of the neural basis of mood disorders in particular and developmental trajectories thereof to identify targets for new treatments, particularly neuromodulation treatments. And essentially what I want to do first of all is just pinpoint, highlight the kind of circuitries that I'm interested in and people in my lab are interested in, and I know that many of you are also interested in, but I just want to sort of start the talk off with highlighting these circuits so you kind of know where we're coming from. Obviously, one circuitry or one complex circuitry that is a very huge part of my work and has been a huge part of my work, as Jeff alluded to, has been the amygdala regulatory affect regulation circuitry. And here is just a snapshot taken from a review article I wrote with my good colleague and friend Holly Schwartz in Pittsburgh a few years ago now, highlighting some of the sort of complexities of this circuitry, obviously the amygdala being the center, medial prefrontal cortical regions in the hippocampus being involved in implicit emotion regulation, and the lateral prefrontal cortical regions highlighted in dark blue involved in explicit regulation. Now that's a starting point, but of course things are much more complicated than that, as we all know. And in sort of more recent years, I would say probably the last 18 months, 2 years, I've become much more interested in looking at the intersection amongst different large-scale neural networks examined with different multimodal imaging techniques, particularly resting state connectivity. And I will be talking a little bit about these shortly. But obviously, as many of you will be all too aware, there are several of these large-scale circuits. I'm just highlighting the three which arguably are the most commonly known and cited and examined. The executive control, if you will, task positive network involved in multiple demands, higher level cognitive executive control. The semantic control, which is involved in switching between the task positive that I just talked about, salience perception and switching between task positive and task negative, and the task negative network, also commonly known as default mode network, centered on medial prefrontal cortical regions, posterior parietal and temporal regions. In a nutshell, as we all know, many of us know, particularly the multiple demand and the default mode network have a yin-yang relationship going on, where traditionally it's thought that this is the case, not necessarily believed by everyone, but it's traditionally thought that the red, if you will, multiple demand network is switched on during a positive task, high levels of executive control. A default mode network is switched off during such contexts, but I'll be talking more about that shortly. And finally, and last but by no means least, a circuitry which probably is more of a centerpiece of my research than other circuits is the complex circuitry reward neural network. And here's just a cartoon of some of the key nodes, obviously centered on the midbrain VTA, ventral, and going to ventral striatum, and then several prefrontal cortical regions involved in different aspects of encoding and evaluating decision-making regarding reward. And I will say quite a little bit more about these regions later on. So the overview of my talk today really is essentially going to be telling you a little bit about the work which we're doing in the lab, focusing on those large-scale neural network developments and how in infancy, and I'm talking about the first year of life, and then early and sort of school year childhood, patterns of activity and connectivity within and among these large-scale neural networks are associated with future depression risk in particular. Then I'm going to switch to talking about multimodal examination of neural networks, particularly reward neural network that we're showing now is conferring risk for mania and future bipolar disorder. And then taking this on to the next stage is using the knowledge of this second area in particular to start to develop new neuromodulation treatments for mania, risk and bipolar disorder down the line, based on our understanding of these neural biomarkers. So let's start at the beginning talking of the first area, which is the work we're doing in the lab focusing on infants and childhood network development and how aberrations or changes, alterations in this large-scale neural network development can actually confer risk for future depression. So first of all, I'm going to be talking about this study, an NIMH funded R01 with my friend and colleague Alison Hitwell, who's a very well-known developmental psychologist here in Pittsburgh. She and I have been interested now for several years in understanding how the infant brain development, and particularly the infant large-scale neural network development, relates to the development of emotional behavior and how caregiver affect might impact those relationships. And just to give you a few pieces of background which kind of motivated our research, as many of you will probably be very aware, that the first few years of life, particularly the first couple of years of life, are characterized by rapid changes in the capacity for emotion regulation. And this parallels the first 2 years of life where there's a rapid development of the human brain. It's known that we can measure and we can assess infant negative emotionality in a variety of different ways, both observationally and behavioral rating scales. And it's known that negative emotionality, so the inability to be soothed, being irritable, etc., is a robust predictor of emotional problems later on in childhood. The corollary of that is that low positive emotionality, particularly infant smiling, joy, laughter, that kind of aspect of behavior. Low levels of that also is bad for you, predicts behavioral inhibition, particularly in early childhood. What is also known is that the caregiver environment is incredibly important during the infant and indeed the whole of childhood years. And we've been interested, and our R01 is really funded to help us understand a bit more about how a caregiver affect and anxiety and emotional instability may impact the development of brain emotional behavior relationships in the first year of life in these infants. So the model we have going forward, and the one that we've been interested in pursuing, particularly in the last couple of years, is this model here, where we are postulating that we have the caregiver affect anxiety environment as a starting point. That obviously, a lot of research has shown that there's a direct relationship between that and the development of infant emotionality, which is not hard to perceive. Obviously, if a caregiver affect is negative, deleterious to mental health, to the caregiver, it's also going to have an impact on the offspring, and that's been shown many times. What we've been interested in doing is bringing in this new piece, which is looking to the extent to which the developing infant brain kind of mediates the relationship between the caregiver and affective environment and the development of infant emotionality. And those are some of the covariates we have available. I have to say this study that we have currently funded builds on a very long history of infant emotional development work and behavioral work that Alison and her colleagues have been performing here in Pittsburgh. Now, the thing I want to really highlight about this study is that we were fortunate enough to be able to look at two independent infant samples. On the one hand, we had our first sample in the 50s of infants, and we examined them at 3 months. And then in the second sample, we had an independent sample from a previous R21, which, if you will, was the run-up to the R01, when we had another infant sample of 31 babies where we could test out our findings in the first sample. And here are just some of the measures that I've just been telling you about. For those of you interested, we use the infant behavior questionnaire, the revised version of the IBQ, to measure the baby's negative and positive emotionality. And then we had all of these measures of caregiver, home environment, state and trait anxiety, depressed mood, etc. So let's just cut to the main findings. For the sake of time, I'm just going to cut to the main chase here. What we first wanted to look at is just to confirm what would be an obvious relationship to first show, which is the direct relationship between the caregiver affect and the infant emotionality. And the key relationship, and this is after controlling for multiple tests, etc. The key relationships that we were able to show was that the greater the postnatal depression, and this is predominantly mothers who were the caregiver, and we unfortunately still live in a predominantly sexist society. So in our sample, it was predominantly the mothers who were the primary caregiver, and therefore those were the ones that were involved in the laboratory and home assessments. So the greater the Edinburgh postnatal depression, the lower this particular measure of infant emotionality, which is a positive emotionality, smiling. Similarly, we showed that the greater the level of trait anxiety in the mother, the lower the level of infant smiling. So that's just a confirmation, really, of what's been shown before, and that's a starting point. If you remember, this would be the base of the mediation relationship triangle. And we also replicated those findings largely in the independent sample, the second sample of 31 babies. Now, the key thing is that we then wanted to look at how the infant brain, the infant resting state, patterns of resting state amongst those large-scale networks were related to low infant positive emotionality, low infant smiling. And in this first sample of the 58 babies, what we were able to show, and let me just talk you through this, is the following patterns, relatively straightforward. What we were able to show, so if you will, if in the warm colors are the positive relationships with infant smiling, and the blue, the cold colors are the negative relationships. What we were able to show is that the greater the resting state connectivity between the amygdala and the executive, predominantly the executive control network shown here, and this is an infant brain template, so it's obviously slightly different from what you might be expected at looking at with the adults. But here we're seeing more positive connectivity, so if you will, a more attempt to regulate the amygdala, the greater the smiling capacity, whereas the opposite was the case with the amygdala connectivity with the salience network and the default mode network. So the more connectivity the amygdala was with the default mode network and salience network, the lower the infant smiling. So this tells us, that essentially the executive function trying to regulate the amygdala may be a good thing for the babies, whereas more connectivity between the amygdala and the task negative default mode network, and also the switching, the salience, perception of salience network, the worse that is for the baby. We were able to essentially replicate the same kind of general pattern of relationships with the positive, negative, and negative relationships, executive control, salience network connectivity with the amygdala in the independent sample. But the key thing that we were able to show was that this pattern of resting state connectivity was a significant mediator between both the caregiver postpartum depression and the caregiver trait anxiety and the infant smiling. So essentially here, I'm showing you the patterns of amygdala connectivity with the whole brain that were shown to be significant mediators of that negative relationship between caregiver postpartum depression, trait anxiety, and infant smiling. Now, I'm going to unpack these in the next slide to show you exactly what this means, but I just also want to point out on this slide that we were able to show largely that these are in fact almost identical patterns of mediation in the independent sample, where we have the same largely two main networks, salience and executive control network, and to some extent the default mode network in the independent sample. Those were mediating this relationship. But the key findings were really with the executive control and salience network. So let me show you the next slide, which was where we were able to, with what we've done here, is just extract from the main clusters of connectivity with the amygdala here to give you a sense of what the mediation actually meant. So let me just talk you through this slide. Essentially here we have the model of the relationship between the caregiver environment and infant smiling, and here we have the resting state mediator that I've just been talking about, infant amygdala, large-scale network connectivity. Now, on this first row, what I'm showing you is essentially the patterns of connectivity between the amygdala and the salience network, and how that was related to caregiver depression on the one hand and infant smiling on the other. So how it relates to here and here. And what you can see here, just going through this top side, we show that the more depression, the greater the caregiver depression, the greater the connectivity between the amygdala and the salience network, but the more connectivity between the amygdala and the salience network, the lower the infant smiling. Similar pattern of findings with trait anxiety. More trait anxiety in the mother, more amygdala salience network connectivity in the 3-month-old infant. The more that was the case in the infant, the lower the infant smiling. Now the opposite was, and by the way, these are alpha and beta representing the A and B arms of the mediation relationships. When it comes to the relationship between the amygdala and the executive network, we have the opposite pattern of findings, as you might expect. So the greater the depression in the mom, the lower the connectivity between the amygdala and the executive network, and whereas the more connectivity between the amygdala and executive network, the higher the infant smiling, and exactly the same pattern with the trait anxiety as the maternal environment. So to cut a long story short, what this is actually telling us is that here we have the caregiver depression, anxiety, trait anxiety. The higher that is, we have a pattern of lower connectivity between the amygdala and the executive network, but higher connectivity between the amygdala and the salience network, and to some extent the default mode network. And that combination of resting state connectivity pattern leads to lower infant positive emotionality, particularly infant smiling. And that was replicated in the independent sample. So as a first study, I mean, this is really telling us that it's incredible, really, that in the 3-month-old infants, we're already seeing these replicated findings in the infant brain mediation between caregiver environments and the infant smiling. And it tells us that, you know, when we're thinking about perhaps targeting babies who might be at risk for future psychopathology, given that low infant smiling in particular is a risk of future psychopathology, that's been shown in a couple of studies, we might want to monitor the activity of these networks, particularly the connectivity with the amygdala, even if we're not going to modulate them with neuromodulation. And so probably not going to want to do that in 3-month-olds, but at least we can monitor their patterns of activity and ultimately use these as ways of monitoring the effectiveness of other interventions. And ultimately, of course, in childhood and adolescence, we might even want to target them with neuromodulation if all else has failed by that stage. So that's kind of setting the scene as to where the large-scale neural network and the integration of the networks is starting to lead us in thinking of risk for psychopathology very early on. And I have to say that what we're looking at now is the interplay of these large-scale networks with each other and infant emotionality, caregiver affect, and also bringing in brain structure, because I've just told you about brain function. We also have some findings which we're now finalizing, bringing in the brain structure. So sort of watch this space, because that's the next piece. So I now want to move on to the second study in this general area, and this is moving on in the developmental lifespan, where we are now looking at children, sort of school-age children, and age 8 through to sort of adolescence. And this is in the context of the Bipolar Offspring Study, or BIOS for short, which is a long-standing study headed by Boris Bermaher and myself, and for now, and shortly, hopefully, will also be Michelle Bertolci coming on, who's very technically skilled neuroimaging data analyst in my lab. But the whole point about this study, as some of you might be aware, is to understand how the developing neural networks, particularly emotion regulation and reward network, and also large-scale neural networks, actually predict the onset of psychopathology risk, by which I mean, you know, sub-threshold symptoms that in turn will ultimately confer risk for and determine and risk for and presence of bipolar disorder. And this is a study in children who already have at least one parent with bipolar disorder. So they already are at familial risk of developing bipolar disorder, but at the moment they don't have the disorder. So we want to know, are they likely to get the disorder? This study has been funded by NIMH very generously now for getting on for 20 years. And early next year, we will find out its fate for the next 5 years or so. I'm going to tell you just about one study which was recently published just last year, actually, and asking questions about the interplay between the large-scale neural networks and psychopathology development. Let me just talk you through the structure of this study. This is a study where we were fortunate enough to be able to follow these children up over 4 years and to perform 2 scans at time 1 and time 2. And at the same time, we're measuring a host of different psychopathology measures including the key symptoms of depression, hypomania, anxiety, affective lability, as well as all the demographic, medication variables, etc., which were controlled for. And the questions we were asking was, can we use neuroimaging to understand where things are going in terms of psychopathology risk? So can we look at time 1 and changes between time 1 and time 2 in neural measures related to these large-scale networks that in turn predict the future, that in turn predict time 2 mania, depression, and anxiety? And the argument here was that, it's okay having all these demographic and behavior measures, but like with the infants, we need hard, objective biomarkers to predict onset or worsening of these subthreshold symptoms that in turn confer risk for future bipolar disorder, if we're ever going to be able to have objective ways of identifying future risk for these disorders, and also targets for treatments. So in this study, we had many different tasks in the scanner, but this is one which I'm talking about today and was the focus of this study, whereby it's a very relatively straightforward emotion regulation task. Many of you will be more than familiar with this kind of task. It's a 2-back working memory task. So obviously, as you would know, people have to remember the letter 2-back and then press the button. That's pretty hard enough. But then they have these emotional phase flankers, which kind of distract you from the task, especially if you're a younger child, and it makes it a little bit harder. So the idea is you're having to bring more of the executive control network online, ideally, to perform this task well, to cope with these emotional distractors. So this is a task which we've been using for several years now in a host of different studies. In the past, we've been focusing predominantly on the amygdala and the task positive network, the executive control network, particularly lateral prefrontal cortical regions thereof. But as I was saying earlier, what we've now moved on to be more interested in is the more complicated relationships between the task positive and task negative network. And here's a copy of the slide from the Tomasi et al. Paper from a few years ago now, showing the yin-yang relationship between the task positive and task negative network during a standard working memory task. So this is where we were looking at. So let me tell you what we found, and I'm just going to cut to the main chase. Now, this is in a group of relatively modest size, but remember, these are children who are gradually getting a little bit sicker. They have a parent with bipolar disorder, and they've had 2 scans 4 years apart. So it's not a really easy study to do, a one-site study. So just bear that in mind. But what I want to show you here is exactly what we found when we looked at this group of children and asked the question, what are they actually doing in terms of task positive and task negative network activity during performance of this complicated task. So here what I'm doing is showing you, if you will, the patterns of activity. In red is the task positive network, and in blue is the task negative network. And essentially what the contrast here is, that the task positive would be doing the 2-back, that's the working memory part of the task, with emotional phase distractors, versus the no working memory condition at all with no faces. So the very, very difficult, or the most difficult part of the task versus the simplest. And the task negative network is the network that is activated for the opposite way around. So it should be activated more to the simple task versus the complicated part of the task. So that's fine. So here we're showing, as expected, the task positive is there as we would expect when the children and adolescents are performing the hard 2-back with emotional distracted 2-back working memory task, and the blue default mode, predominantly default mode task negative network is there when they're not. But if you look carefully, and we sort of try to decompress this and try to work out exactly what was going on in a slightly more sophisticated way. What we're showing here, let me just focus first of all on the task positive network, and this is in dark colors here. And this is, so here, if we look at on the right-hand side of the graph here, what we're showing is that this task positive network, and here I'm showing you the host of different regions, is, as I've just shown you, switched on, activated during performance of the 2-back, an emotion condition, emotion distractors. What we're also showing now is that it's actually deactivated. This task positive network is actually deactivated during the opposite, the 0-back condition. Whereas the task negative network, now shown here, During performance of the hard part of the task, the two-back with emotional distractors is deactivated. But interestingly enough, during the easier part of the task, it's not as if this network is activated, it's just deactivated less. So the story is, the task positive network is truly activated really only during the two-back. But the task negative network is deactivated, should be deactivated to a far lesser degree during the zero back, the easy part of the task. So it's a story of deactivation. The take-home message that this task negative network should be truly deactivated during performance of the two-back task. So bear that in mind, because what I'm going to show you next is what happens if it's not, or if it's not deactivated as much as it should be. And this is the key, one of the key findings we found. So here, I'll just go back a stage. This is part of the task negative network, which should be switched off during the hard part of the task, the two-back with emotional distractors. And here it is again. What we found, and this is after controlling for multiple tests, I'm not going to sort of bore you with all the statistical details here, but essentially what we found is that this was the pattern. The lower the level of deactivation in this network, which should have been deactivated during performance of the two-back task, the greater the depression 4 years later in these children. So this was at time 1. So the lower the level of deactivation, the less switched off than it should have been, the worse off the children were. Now, this is obviously not a linear relationship. It was a very much a non-linear relationship, as you can see. And these are the raw data. This is the fitted curve. What we then asked the question about was, well, what happens between the connectivity between this lack of deactivation in the task negative network and the task positive network? What happens if the lack of deactivation is this lack of deactivation somehow interfering with the task positive network? And is that what's really causing the problem? And indeed, that was the case. Because what we also showed, and again, this is controlling for multiple tests, etc., was that the greater the connectivity between this lower than should be deactivation in this mid-cingulate gyral region in the task negative network and connectivity with the task positive caudate nucleus, and that's caudate activity is known to be part of the working memory network, as I'm sure most of you will know. The more connectivity between that during the difficult part of the task, so the more, if you will, interference by the task negative network with the task positive, again, the more depression at time 2. And this was actually a change in connectivity from time 1 to time 2. So it wasn't just what we're looking at time 1, it was an increase in connectivity from time 1 to time 2. So it really is suggesting that it's not so much what's happening in each network, it's how they're talking to each other, how they're connected and how they're interfering, which really does seem to determine worsening of symptoms that we know confer risk for depression and bipolar disorder later on in these children. So this is something we're now following up in larger samples, obviously. So now I want to switch to the second major part of the talk, which is really asking the question, okay, we can look at and use multimodal imaging in various different ways. And I've shown you 2 examples of what we're doing in developmental work in the lab. But now I want to really focus on the multimodal studies of what we're doing in another part of the lab's work, which is very much looking in much more detail at reward network function and how multimodal examination of reward network function and various intricacies thereof actually confer risk for future mania and bipolar disorder. I'm just going to start by telling you a few home truths about bipolar disorder, and apologies to those who are all too familiar with this, but I just, I always find it surprising how many people, including some of my esteemed colleagues, still feel that bipolar disorder is a bit of a Cinderella psychiatric disorder. It's not really that important. I just want to highlight these facts, because I think these suggest otherwise. It's common. It's very common. It's 2.6 12-month prevalence, nearly 5% lifetime prevalence, even common in adolescents, let alone, children. So, it's not something to be ignored. it's characterized, obviously, as we all know, by mania and depression, but it's the mood swings and the inability to kind of control the switching between these mood states, which of course is the debilitating feature. And they're just not knowing how to control or regulate your mood is just devastating. I have to say, several colleagues and a couple of close colleagues at the moment who sadly suffer from bipolar disorder, and it's tragic to see them not be able to regulate their mood at times when they really need to, and it's impairing their career. And of course, with other people, it gets far worse, as I will show you in a moment. Mania, I just want to highlight, of course, mania is the characteristic symptom of bipolar disorder, and we all know that. But when we want to study bipolar disorder, it makes sense to understand factors, mechanisms that confer risk for mania. Because of course, understanding risk for mania also will help us understand risk for depression because of the switch mechanism, which kicks in, as naturally, if people are not medicated, they switch from mania to depression. I remember when I was a medical student, my first bipolar patient I saw was almost psychotically manic one week, and the next week, without being on much medication, he slipped to almost psychomotor retarded depression, and it's quite shocking to see. So it's almost if the brain gives up and then exhausted, and then you switch from a mania to depression. So the bottom line is understanding mania risk is key, I believe, to understanding mood switching and ultimately bipolar disorder in general. Just to point out, it's the fourth leading cause of non-communicable neuropsychiatric disability in the world. It's not trivial. And of course, the devastating facts are here. It takes about 10 years off your life if you're unlucky enough to have it. 20 to 30 greater suicide risk than the general population. In fact, it has arguably the highest suicide risk of all psychiatric illnesses. So isn't it surprising and shocking that we haven't come far enough? We haven't made more progress in understanding the brain basis of bipolar disorder, because until we do, we're not going to be able to really help develop new treatments. Now, of course, it's a very complicated disorder because it has so many different phases. It's got a double hit, mania, depression, and then the mood swings in themselves and just general emotional dysregulation. It's a devastating illness, but even before the psychosis kicks in. So, this is why I'm passionate about studying this illness. And I just want to also highlight the fact, and again, apologies for preaching to the choir here, but a lot of people tell me, well, we know how to treat mania, we know how to treat bipolar disorder, but do we? I mean, there are lots and tons and tons of medications that we use, but as we all know, sadly, many people with bipolar disorder often need to be on 4, 5 or more medications to even keep well. Again, some of my colleagues are taking 4 or 5. They don't always work. They have unpleasant side effects, to say the least. They're not always tolerated, lithium being a prime example, don't always work. And even if people are relatively stable, they're still at risk for relapse into mania and depression. And then once they relapse, they have to change their medication, go through all of this again, and so on and so forth. So again, I pose the question, if only we could understand the neural biomarkers that confer risk for mania, that would help us understand risk for mood switching and depression risk, and help us ultimately understand risk for bipolar disorder, as well as providing targets for new treatment. So this is what we want to see. We want to see a diminishing of the risk for mood switching, mania and mood switching. And this is kind of where we're focusing. So where do we start? Well, it's easier said than done, isn't it? Trying to understand the neural basis of risk for mania, because mania is kind of complicated. It's a complex structure in itself, a complex series of symptoms. So I want to sort of highlight the fact that there are clues in the large psychology literature in bipolar disorder, headed by my colleague and friend Sherry Johnson over in Berkeley, and others, Laurie Alloy being another, that have indicated quite convincingly that there are certain behavior traits or response tendencies, if you will, that confer risk for predisposed to bipolar disorder. And these are high impulsivity and high sensation seeking. And just to highlight what these are, the impulsivity, which in of itself is a complex series of different behavior traits, as we all know, is rash behavior, basically, little forethought or consideration of the consequences. It in of itself has various different components. These are all prevalent and on high levels experienced to a high degree in young adults who are at future risk for bipolar disorder. Sensation seeking is another behavior trait which also is there present in individuals who are at high risk for future bipolar disorder. And this, of course, is the tendency and willingness I think the emphasis being the willingness to take risks for novel and intense sensations, even though, you know, there may be a cost to that. So, as I just said, that been shown to actually predispose to hypomania in young adults, but also, of course, evident in adults already with young adults already with bipolar disorder. So constraining our search, if you will, to these behavior traits, I think is a very promising way, because it tells us a lot about the journey to mania, and therefore mood switching and depression and bipolar disorder risk down the line. How do we measure it? Well, I have mentioned this task before that I'm about to show you, but I just want to go through it once again, because I want to highlight this is a task, by the way, a reward task, which was developed by Erica Forbes, Henry Chase, and myself in Pittsburgh, based on an earlier version of this task, which Erica had herself developed. This is a reward task which is kind of built on the MID task, and it essentially contains an expectation phase and a receipt phase. But the reason it's important, and the reason we designed this particular version, is that we really wanted to hone in on the reward expectation or the anticipation phase, because it's during reward expectation that frustration is triggered in people who are more impulsive, more reward sensitive, more sensation seeking. If they're in a context where they may or may not get a reward down the line, that's frustrating, that's irritating to them, and irritating to anyone who's, you know, more impulsive, because you want to move on, you want to get the reward, you want to move on, you know, you don't want to wait. So it's very frustrating, and that's it's in those sorts of contexts when frustration is kind of triggered, that impulsive acts and all those deleterious behaviors that we associate with mania can be triggered as well. So that is why looking at reward expectancy as a phase in reward is, I think, particularly important when we're looking at risk for future hypomania, mania, and ultimately bipolar disorder. So very briefly, let me talk you through the structure of this task. This is a card guessing task. So basically at the beginning, people are shown this symbol, which means, you know, please guess whether the card you're about to see in a few seconds in the future is going to be higher or lower than the number 5. Okay, so this is one of 4 contexts that they see. Arrow up means if you get the answer right, you're going to win money. If you get the answer wrong, don't worry, nothing will happen. Arrow down during the expectancy phase. If you get the answer wrong, this time you're going to lose money. Answer right, nothing will happen. No money won. Up and down, high risk situation here. If you get the answer wrong, you'll lose. If you get the answer right, you'll win. And then we have a neutral control when nothing happens regardless of what you do. To cut a long story short, this allows us to compute a parametric regressor, so we can actually kind of assess the extent to which there's greater or less expectation of win. So obviously this is high, there's a 50% chance of win. Here there's 0% chance of win, there's a 50% chance of loss. And here it's less, because although there's a 50% chance of win, there's all that's offset against a 50% chance of loss. So we can gauge by sort of parametrically modulating the expectancy value of reward, in other words, the probability of future reward happening, we can actually assess the extent to which the reward circuitry actually is ramped up or ramped down according to the extent to which reward is expected or not. And just to cut a long story short, early on, using a slightly earlier version of this task, we were able to show in a study, and I was partly consulting in the University of Cardiff in the UK at the time, and this was by Xavier Caceres there. What we were able to show is using this task that people with bipolar disorder, both bipolar 1 and bipolar 2, when euthymic, so not yet hypomanic, showed elevated activity as we had expected in ventral striatum, but also perhaps slightly unexpected in this large area centered on the left ventrolateral prefrontal cortex, which I'm going to talk more about shortly. At the same time, back in Pittsburgh, we had the same task with a different setup, a different group of subjects, people with bipolar disorder who are currently in depressed mood, people unipolar depression, currently depressed, and healthy controls. But again, the same findings. So even though there's completely different people and different, in fact, a different continent, different scanner, same task though, we were able to show that people with bipolar disorder showed this elevated pattern of left ventrolateral prefrontal cortical activity during expectation of reward, relative to depressed unipolar individuals and healthy controls. So that was a starting point. We also had some other findings as well, which I'm not showing you similar pattern. Okay, so the left VLPFC was there during expectation. Next question, as I was just saying earlier, you know, we've looked at people with bipolar disorder, albeit not in the manic phase. Obviously, it's very highly difficult to get people in florid mania to lie still in the scanner, let alone do this task. But we went back and said, well, what about people who might be at risk of future mania by virtue of having high levels of impulsivity or sensation seeking, fantasy seeking? Are they showing the same pattern of elevated reward circuitry, elevated left VLPFC activity during this task? And the answer was yes. So again, here I'm showing you, this is a group of about 100, I think it's about 102, 101 young adults, 18 to 25. Henry Chase and I led this work in my lab. And this was asking the question, these young adults, we're recruiting them in an NIMH-funded study across a whole range of risk of bipolar disorder and mania by virtue of their levels of impulsivity and sensation seeking. And what we did is we derived, if you will, a complex trait, a sort of a summary factor, which comprise different facets of impulsivity, particularly urgency and sensation seeking, and regress that on the whole brain. So this is a whole brain, non-a priori ROI. And here we have very larger than life here, left VLPFC, more increase in activity as the probability of reward went from 0 to high during that, or 50% in that expectancy phase. And that was a positive relationship with this impulsivity, sensation seeking. So the more impulsive, the more sensation seeking, the more likely you were to show this pattern of elevated left VLPFC activity. Also ventral striatum as well. Now, we then sought to replicate this finding in an independent sample of 127 young adults, and indeed found this. And this is work led by Carol Edenston and Jay Fournier. Jay subsequently moved to OSU, but still actively collaborating with myself. And again, we were able to show this relationship. Now, this is using an ROI approach because we had a complex modeling, as I'm about to show you. But what we were able to show is that in a completely independent sample, just looking at the relationship between this left VLPFC and other regions in the context of the reward expectancy, that increasing from very low 0 expectation of reward to 50%, we were showing that those with higher levels of impulsivity, particularly negative urgency, this a key feature of impulsivity, the more on the urgency, the higher the level of activity during reward expectancy. And here is the combined sample of 227 young adults. The reason it was a complex model was that we also went on to look at a mediation, because what we really want to know is, does the left VLPFC heightened level of activity during reward expectancy actually predispose to future mania and bipolar disorder via the negative urgency? So it's is it that the brain predisposes to this behavior trait? They're probably highly bidirectional relationships, quite frankly, but is there a relationship between these two that in turn predisposes to mania risk? And here, as you can see, we use this scale, the mood scale, which is a wonderful scale, if you're not familiar with this, developed by my esteemed colleagues, Ellen Frank and David Kupfer, several years ago now, over about 20 years ago, with colleagues in Italy. And this is a wonderful scale, although it takes a little time to administer, because it actually assesses in depth all the subthreshold, subclinical, if you will, tendencies that relate to future mania or depression, i.e. bipolar disorder risk. Much more sophisticated, much more subtle way of examining in depth than, say, the YMARS, the mania rating scale, or other measures purely of mania. So we use this, and this is one of the many factors that we were able to show. I mean, I have other factors that I could have shown you today, but this is just one that is particularly shown to be a risk for mania and bipolar disorder. Anyway, what we showed was that negative urgency does indeed mediate the relationship between left VLPFC activity during reward expectancy and future bipolar disorder risk in this way. So the more activity here, during reward expectancy, with increasing expectation of reward, the greater the negative urgency, and that in turn predisposes to more likelihood of risk for mania down the line. We have subsequently replicated that yet again in another sample of 103 individuals, some of whom are included in that previous sample, but not all. What is different about this sample is that we were able to measure the moods risk over the course of a year. This was measured just a snapshot in time. What this is doing is a longitudinal relationship. So here we have a mood. This is a more even more general measure than the mixed instability that I showed you before. This measure. What we're showing you here is the manic domain. So this is actually pure mania risk, if you can call it that. It's an actual very accurate measure of mania risk down the line, lifetime mania risk. And again, this is measured over 3 time points: baseline, 6 months, and 1 year. And we're showing that the higher activity in the left ventrolateral prefrontal cortex during reward expectancy, the higher the risk measured 3 times in this group of 103 people. Now, moving on, because I'm appreciative of time, I just want to highlight the fact that, of course, we're not just stopping at fMRI. We're also using EEG. As you all know, this has higher temporal resolution. And Dean Salisbury, who's my colleague, who's an expert in the use of EEG, as I'm sure you all know, he's kindly agreed to be photographed, showing how the setup works. Interestingly, with EEG, what we know is that it's high sensitivity, particularly over the VLPFC. The VLPFC is easy to measure in terms of underlying EEG activity, but it's got a relatively thin intervening tissue, and also the orientation of the pyramidal cells in the layers in that region are suitable for tapping into with EEG. We also know from other studies that EEG power in the beta band in particular is associated with reward expectancy. And this in turn reflects underlying dopaminergic activity. And if you remember, and as obviously most of you will be more than well aware, dopamine is one of the key neurotransmitters in reward circuitry, from midbrain to cortex and also to striatal regions. We also know that beta power in prefrontal cortical regions in general is related to sensation seeking in a positive way. So more beta power in prefrontal cortical regions during reward processing, more sensation seeking. There's been a couple of studies, not that many, but a couple of studies showing that. So we kind of wanted to put this all together. And in a snapshot, I'm just showing you what we found. And this is a study just published earlier this year. This is Brian Kaufman, who's a very talented junior faculty who works in Dean's lab and also with me. And what we were able to show is using our task, honing in on the VLPFC, a relationship between that and sensation seeking. And let me just talk you through what we found. So in this, we have the possible win. So 50% chance win. Here we have, you could win or you could lose. Here's neutral and here's the loss. So, high chance of reward, no chance at all of reward. The opposite, in fact. And what we showed here is that only at all do we find left ventrolateral prefrontal cortical activity in the beta frequency? This is sort of high beta frequency gamma range here. Did we find that there was activity over the left VLPFC during this reward expectancy context? In the other 3 contexts, there was not there. What is more is that left VLPFC beta power during this possible 50% chance of win context was positive related with sensation seeking, particularly thrill adventure seeking. This is the Zuckerman sensation seeking scale for those of you who are interested. So again, we're showing a multimodal approach to understanding mania risk. Now, I just want to skip through these next couple of slides because I think it's important to ask the question, what is the left ventrolateral prefrontal cortex actually doing? I've highlighted it as a key area, but what is it actually doing in the context of reward? So here, I want you to think about a scenario in which you have an opportunity to win a large or not so large reward. And what you've got to do is you've got to choose between a red circle or a red square. And down the line, you'll find out whether it's the red circle or the red square that will lead you to the big reward that you seek. What you subsequently learn when you start choosing and you're getting feedback is that the red circle, most of the time, has a high chance of leading to the green triangle, which in turn is going to give you the large reward. The red square, on the other hand, most of the time gives you a high chance of this purple triangle, smaller purple triangle, which gives you much lower reward. So what you've got to learn is the contingency, the relationship between the red circle and the green triangle and the red square and the purple triangle first. It's not that this gives you reward, it's the relationship with the green triangle. And it turns out that the left ventrolateral prefrontal cortex does exactly that. That's what it's doing. It's actually making associations between two otherwise neutral stimuli that in turn will allow you to optimize future reward. And this is a study, cognitive neuroscience study performed in the University of Oxford by my friend and colleague Tim Baron's lab, showing just that. So essentially the left, and this is, by the way, apologies, this is the left ventrolateral prefrontal cortex, almost identical region to the region we show. It's just switched in the orientation in their paper. Essentially, the left ventrolateral prefrontal cortex is making decisions and determining the contingency between two stimuli that in turn predict optimal reward. So that's kind of good. But on the other hand, we know that the left ventrolateral prefrontal cortex doesn't necessarily always denote good things. So here's another scenario. Imagine you have to choose between these two reward options. So this is again a reward situation when you can either optimize, you can have a high or low reward, but this time rather than a probability or an amount, it's a delay. So you can have a small reward now or $100 a large reward in a month. What are you going to choose? Probably most people would likely choose $10 now because a month seems a long time to wait. But if it were $1 now and $1,000 in a month, you might wait. And that's what most of us would do. People with bipolar disorder tend to make more impulsive choices, so they're much more likely to go for the smaller reward now, despite variations on the theme. And what has been shown by a very nice meta-analysis published a couple of years ago now is that a region of the left ventrolateral prefrontal cortex, not a million miles away, very nearby the focus that I was just showing you, is actually involved in promoting this immediate reward choice. So in essence, the left ventrolateral prefrontal cortex, more active, tends to promote the choice of immediate small rewards over large rewards during decision-making contexts such as these. And essentially, we can ask the question, how is impulsive choice measured? And I just want to highlight how it is measured, because I'm going to come to this in the next slide. What I'm showing you here during a delay discounting or intertemporal decision-making task is a time delay in months. So you can have 0 time delay, 1, 2, 3, 6 months. This is the time you're going to have to wait if you're going to choose the large reward. And on the y-axis, I'm showing you that the relative value of the large relative to the immediate reward. And this is the discounting factor effectively. That's where this comes in. So the more you discount the large reward, the less likely you're going to choose the delayed reward, the more likely you're going to go for the immediate reward now. Now, obviously, as time goes on, most people drop off and say, okay, I'm going to just go with the immediate choice. And if you're particularly impulsive, you're going to go for the immediate small rewards sooner rather than later. So here is the likelihood effectively of choosing the large delayed reward. So as the time gets longer and longer and longer, zero, it's of 0 value, you're not going to choose it. So if you're impulsive, that drops off quite quickly. If you're less impulsive, you might be more patient and you might be more prepared to wait. So at 3 months, it's not so it's not so useless to wait for it's a 50% chance that you're going to wait for it. It's of that value to you. Whereas if you're more impulsive, you're not. And this is the K, what's known as the K function. You can derive the K value. I won't give you the formula now, but it's basically a measure of delayed reward discounting. The larger the K, the more impulsive you are. So hold that fact, because what we then did is we designed a study where we can essentially measure the impulsive choice, if you will. And this is a task derived from many other delay discounting tasks. We used it so it's compatible with both fMRI and EEG, which I'll show you in a moment. But essentially, it comprises 2 main components. One is when you present the 2 options. So here we have the immediate reward in pink versus the delayed reward in blue. In this case, it's not a difficult choice. Probably most people would choose the $20 now, because waiting 14 days for $10 more probably isn't worth it. But if that were $1,000 in 14 days, you'd probably go for that. The point is we show that, and then we get people to mull it over, and then we show them a choice when they now have to sort of put their finger where they want to go, and then they choose either the pink, which is the immediate, or the blue, which is the delayed. So this gives us a window where we can measure brain activity during the decision-making phase. And essentially, and this is just one slide I'm showing you, this is preliminary data. And I'm pleased to say that we've just found out that we're getting funded, a nice foundation, Milken Institute is funding us to do a study using this task with EEG and other things, which I'll talk about very shortly. Bottom line is that we find that people with bipolar disorder on the right, when they're during the decision-making phase, just before they choose immediate choices, they show greater activity in the left ventrolateral prefrontal cortical region. And this is, of course, whole brain analysis. What is more, they have a higher K, as shown here. This is obviously very small numbers. It's a pilot study, and it's proof of concept, pilot data for the grant that we thankfully and gratefully have got. So what this tells us together is that adults with bipolar disorder, they have greater left ventrolateral prefrontal cortical beta power when deciding between the reward options, and this appears to predispose them to choosing immediate small rewards more than in healthy controls. So let me get on to the final piece of my talk, and really, I'm just going to give you a snapshot of where we're going with this. And this, we're very excited about this. Can we use all of that which I've just said in the context of bipolar disorder to lead to new neuromodulatory treatments? Badly needed. If all we do is prevent people from having to take 5 medications, they can get down to 2 or 3, that surely must be a good thing. And hopefully we can do more than that. So what we did in this field is ask the question, can we target the left ventrolateral prefrontal cortex and underlying reward circuitry connectivity with ventral striatum, et cetera, with a first type of neuromodulation, direct current stimulation, TDCS, transcranial direct current stimulation for short. And this is just the first type of neuromodulation that we've used. And this was an R21 funded by NIMH. And we finished the study in about 30 or so bipolar individuals relative to healthy controls. And essentially what I'm showing you is the one slide, the main findings, and this was published a couple of years ago. Essentially what we did, and the advantage of TDCS, despite its other disadvantages, which I'll say briefly in a moment, we're able to administer this actually in the scanner. So we're actually administering it when they're doing the task in the scanner. And what we were able to show is that when we were administering the TDCS, we were trying to downregulate the left VLPFC activity with cathodal TDCS. The lower activity during the stimulation, during reward expectancy in the left VLPFC, the lower the post-scan negative components of negative mood to do with arousal and irritability, not depression, but irritability. So this was the PANAS scale, and we were looking particularly at the items that were to do with anhedonia, depression versus the arousal, irritability, and it was very much the arousal measures that were reduced. So this is a first start telling us acutely that by lowering left VLPFC activity during reward expectancy, we can have an impact acutely on the mood. And of course, irritability, arousal is very much part of the mania syndrome, as we all know. However, of course, as we also all know, TDCS has its moments, it also has its disadvantages. And a problem with TDCS is not terribly focal. Some would argue it's very weak, it's very diffuse. Are we really hitting the area we want? So this brought me to think more about using theta burst stimulation. And this is very much in collaboration with my colleague and friend, Fabio Ferrarelli in Pittsburgh. And Fabio is an expert in the use of TMS. And we together have, very thankful to NIMH, recently been funded with an R01 to look at the extent to which we can target the left VLPFC during reward expectancy, particularly measure its activity pre and post TBS targeted to the left VLPFC versus to a control area, the somatosensory cortex. And here's a pilot data which we had for the grant application, and just showing you that actually we were able to show that in people with bipolar disorder, there was quite a remarkable pattern of reduction in connectivity between the left VLPFC and ventral striatum during reward expectancy as the stimulation went from the control to the one that we're interested in, left VLPFC. So essentially, the left VLPFC reduced this connectivity between the left VLPFC activity and the ventral striatum activity, much more so in individuals with bipolar disorder relative to healthy controls. And finally, I just want to show you pilot data which we had for the foundation, the Milken Foundation grant, which is now funding us to do the EEG work with neuromodulation. The same idea, but this time with a different task and with EEG, and a different, some different montages as well. But essentially what we want to do is examine the extent to which targeting the left VLPFC with inhibitory TBS, again, continuous TBS to the left VLPFC can actually damp down activity in the reward circuitry and subsequently mood related with mania risk. And indeed, our pilot data shows that that is indeed the case. So here I'm showing you pre-CTBS, individuals with bipolar disorder have elevated beta power over the left VLPFC relative to healthy control. After the TBS, it's reduced. And this, of course, is the left VLPFC specifically during the reward expectancy condition. So in summary, I'm very pleased to have presented a little snapshot of the work we've been doing. And I really want to highlight, there's a lot of things still to do, of course. We've been looking at interactions among the amygdala and large-scale networks and how this predicts concurrent and future depression risk across infancy and youth. And as I said earlier, we're following up these studies now with more data, larger samples, etc., longitudinal samples as well. That in turn is going to lead to targets for neuromodulatory treatments or monitoring of other treatments. Multimodal imaging in bipolar disorder is kind of a bit further along. We've already identified some markers, particularly the left ventrolateral prefrontal cortex, both with fMRI and EEG, and now are using these to develop new neuromodulatory interventions to really tackle the problem of needing better treatments, better interventions for bipolar disorder. And a big plea, as I keep on saying, as a clinician, I've been so demoralized by having, seeing patients have such a dreadful life because of the medications that keep having to be switched 5, 6, sometimes more, different over the lifespan. Wouldn't it be lovely if we could just take people away from all of that and give people a chance to have a treatment that works, maybe in tandem with a couple of other medications, maybe with psychotherapy, but at least give people a chance to get better, to have a lower risk of becoming manic without the need for a load of different medications, often which don't work. So at that point, I just want to say thanks to my lab. This is a picture taken of my lab prior to the pandemic, I want to say, at a party just actually about 2 weeks before we had to shut down. Little did we know we'd have to shut down during that party. And hopefully we're going to be able to have a party like that again soon. They're a wonderful group of people without whom, of course, I couldn't have done any of this. And IMH have been incredibly generous, and here's the funding of which I'm PI, the Pittsburgh Foundation, and of course the BBRF, and to come will be the Milken Foundation, and I'm very grateful to all of them. I also thank you so much, very, very grateful to all of you for taking time out of your busy schedules for listening here today. So thank you again.
Mary, that was really amazing. What a tour de force. Thank you so much. And what a great note to end on about, hopes for the future and reducing polypharmacy and morbidity and mortality from these very serious illnesses. And it was great that you sort of reviewed for us, many of whom I think some are attuned, but like the real impacts of these conditions. And it was really lovely to hear work from a developmental, I think what gives us hope in listening to you speak is like the developmental perspective and the idea of earlier intervention, right? Before someone has had many episodes of mood episodes, many medications that are added one onto another. And I think the earlier we can identify risk and intervene, I think that gives us real hope for the future. So we've got questions that are coming in through the Q&A, and I would encourage people to click on the Q&A button and add questions to them. I'm going to start with a question from Jacques Ambrose, who says, I was wondering if Dr. Phillips has any thoughts on the cultural variables, this is referring, I think, to the first part of your talk, in infant caregiver interactions. For example, in Pacific Islander and or Hispanic communities, infant caregiving is shared among multiple of members of the family, higher rates of intergenerational cohabitation and communal raising. Is there a meaningful difference between repeated interactions with one primary caregiver versus multiple shorter interactions with multiple caregivers?
Very, very great question. Very interesting perspective, actually. Yeah, it'd be great to look at that in more detail. You know, I think our work suggests that it's the amount, the quantitative amount of positive caregiving environment. And, you know, I think even outside the Pacific Islanders, there are other cultures, indeed within Pittsburgh, whereby you know, often grandparents are taking on some of the burden of the care, well, the joy of caregiving, as well as parents. Often the parents are out at work; they can't be there. I know when I was growing up in the UK, that often was the case. Some of my friends were brought up by grandparents and so on, as well as parents. And I think as long as there is the positive environment there, I mean, whether it comes from just one person or two people, I don't think, you know, I think we don't know for sure how it's going to impact the infant brain because we haven't studied that yet. But I think what our research tells us, it's the total amount. Now, there's obviously aspect of attachment which one can get into there, and again, you know, having a key attachment figure is going to be important, but it doesn't have to be one person. And indeed, even in our studies, we had to, for the sake of ease, have one caregiver, the primary caregiver, and as I alluded to, it's usually the mum, but not always. In fact, we had a couple of dads who were the primary caregiver in the 1221. And as long as it's someone who's giving you, no matter how many people, I think that's the key thing. It's the kind of amount of environment. And, you know, I think what's astonishing is that the infant brain is pretty resilient. So, you know, even these poor little babies with very negative caregiving environments are still there, still able to have some level of infant smiling. So it's not as if you've got to have one parent who's really happy all the time and then you're fine. You've got to, you know, it's the amount and the quantitative amount, I think, I suspect. But that will be a fascinating thing to study. And indeed, you know, Alison and I have often talked about, wouldn't it be great to look at dyad, you know, or that intergenerational, you know, caregiving effects. So great question. We're on it.
Great, thank you. The next question is from John Mann, who says, "Great talk, Mary. Any concern that anti-manic treatment might increase risk of depression?" And I assume that he's referring to the neuromodulatory work that you presented toward the end of the talk.
Absolutely, great question, John. Thank you for that question. You know, that's something we obviously are well too aware of. I think, you know, the point we're coming from here is that, yes, of course, it could flip people into the opposite pole, but I think that's probably less likely. I think because given the nature of our treatment, we're not trying to get rid of any activity at all. We're trying to dampen down a heightened level of activity. Now, of course, that's not to say that it might have downstream effects down there. And you know, there could be some people for whom it's just not appropriate. But we do have options of ramping up and ramping down. I mean, in terms of amounts, at the moment, it's just acute. So it's just a one-off, but of course, down the line, we want to do a trial when we have repeated sessions, and some people may need less, some people need more. The threshold for some people may be less than the threshold that we're currently giving. We're usually giving, like, you know, like 150% above, you know, motor threshold, and we might want to ramp that down for some people. You know, I think we just need to show that, and we are hopefully already starting to show that this is working, at least in our TDCS, and we have no reason to suppose it's not going to with TBS. But if we do find it's too drastic, we can dumb it down a bit. We can dampen it down. Of course, the other thing to say is that I'm not suggesting that the neuromodulation alone, that's it. They're going to have that and then they'll be cured and they don't need anything else. No, of course not. We would need that alongside perhaps a mood stabilizer or an antidepressant, or perhaps, you know, a new generation mood stabilizer, ideally, and psychotherapy. All the things people are having at the moment, but just not so much of the psychopharmacology load. I mean, that is what we're trying to do.
Yes. Great. This is a question came in from a trainee, Greg Gabrielis, sort of dovetailing on what you just said about, you know, questioning different therapeutic modalities for bipolar disorder. So Greg said, "Hello, and thank you for your talk. I think that your approach to look for biomarkers for the, quote, bipolar personality is interesting, but I'm not clear why you assumed that the therapeutic implication is neuromodulation. The brain seems like it is a social organ and changes with the social environment and in psychotherapy. If a lot of the reticence among patients is fear of losing the creative aspects of bipolar disorder, then won't directly adjusting the brain play into those fears? Wouldn't looking at prevention through making the social environment healthier or changing the brain through integration and psychotherapy also be approaches?" So, and I think you've already started to address in your response to John that you don't see this as mutually exclusive, but exactly. I'm curious if you have other thoughts or responses to Greg.
I think that's a really fair point. Greg. And you know, I'm a psychiatrist, and I certainly don't think that, you know, medication or shall we it's direct intervention in the brain, if you want to say, is a be all and end all. I mean, but you know, the thing you have to remember is that what we're showing is that here we have a predisposition. What we're looking at is really the brain-based basis of a predisposition to future mania or ultimately bipolar disorder. We're looking, if you will, at the brain basis of the behavior trait, almost like the personality, not it is the personality, but it's the behavior trait, the response tendency, which is bad for you. You know, no matter how you come across this, you know, mania, predisposition to mania is not good for you. You know, you might think about it as being creative, but it's not great for you. And we are going one stage before then. We're looking at the brain basis of the behavior trait. All we're trying to do is diminish the risk of that behavior trait leading you down the garden path to trouble. We're not saying we want to get rid of it at all. Of course not. I mean, there are people with bipolar disorder with this high level of impulsivity and sensation seeking will always have this tendency. What we're trying to do is stop that tendency from being manifest in ways that are dangerous. We certainly do not want to take away creativity. So I think of neuromodulation as more like an adjunct to psychotherapy, actually. I think of it as a way of just changing the way the brain might work a little bit in different contexts without taking away its capacity to be itself. And indeed, I mean, this is how psychotherapy works. The psychotherapy is telling you to, right, you've got a tendency to be a bit irritable, a bit frustrated in context, regulate yourself, regulate. I mean, that works via the brain. You're changing the brain with psychotherapy. I mean, you're not changing it via the kidney, you're changing it via the brain. So all we're doing is a slightly more direct way of helping psychotherapy work. I mean, this is what we could do. I mean, we might be able to reset the brain basis of the response tendency so that psychotherapy has a better chance of working. I mean, that's another way of thinking about it. But I take your point, and I think all these things work together. As I keep on saying, you know, I do not want to see patients of mine be on 5 medications, none of which really work. And what, you know, and then they get sick, and then the dose of one goes down, and then everything's up right, and then they get all the side effects. I mean, that's what we're dealing with. That's the reality. We've got to do something better than that.
Thank you. So Jay Gingrich had a question that was very similar to a question of mine. So I'll ask his, and I'll append a little bit to the end of it. So Jay says, brilliant work. How can you disambiguate the effect of mother's mood? I think so. He's referring to your first, the first piece of work that you presented. How can you disambiguate the effect of mother's mood from inherited genetic factors that could be influencing a child's development? Were any of the mothers in your developmental study taking antidepressants or anxiolytics? So if I may just add sort of related questions to it, you know, the mother's emotional measures that you reported, one seemed like more of a state measure and one more of a trait measure, right? One was depression severity and the other was a trait anxiety measure, right? And so I guess I wondered about heritability between those, the resting state functional connectivity, and then the expressed emotion of the infants, and how sort of, right, how modifiable those things are, how much reflects, this sort of intersects with what Jay said, how much reflects genetic heritability versus environment?
Yeah, yet another great question. You know, we've been trying to look at this. In fact, we put in the ground, which didn't get discussed, and looking exactly at this, the intergenerational and transfer of neural network function and inherent emotional behavior. Now, for some reason, the reviewers didn't like it, so draw your own conclusions. But of course, it's very hard to disentangle the effect of caregiver environment per se upon the genetic basis for the caregiver having a predisposition to depression, which is likely to be inherited by the child. Of course, we appreciate that. Yeah, so this is we think of this as a first stage, showing that we can have quantify the caregiver affect and anxiety, and actually look at those whilst controlling for or not controlling for other aspects of the caregiving environment, such as the stability, you know, of the environment, the poverty, the extent to which the parents engaging, the caregiving style, all of this kind of stuff. So, you know, whilst we're not looking at genetic factors per se. We're not looking at the parent's brain and how that is activity is similar to the baby's. What we are trying to do is covary out other factors outside of the affect and anxiety per se that might be contributing to the baby's pattern of resting state activity. So we did actually include, as you might remember, with the home variables, which are measures of the sort of caregiving style and environment generally, and then we have poverty. These are not the same as the intergenerational risk, but they are kind of symptoms of their factors related to. Now, interesting enough, the reviewer of the biological psychiatry paper didn't like it when we put in those home variables. So we did analysis with and without, and actually the findings were very similar. So I think we can be confident that our findings are more to do with current caregiver mood, whether it's current depression or trait anxiety. And interesting enough, we did look at state anxiety, affective lability, other measures, and it's really only the postpartum depression and trait anxiety where we found these significant findings. Next step, absolutely. I mean, in fact, what we would love to do is indeed follow up from the grant that didn't get discussed and look at the extent to which patterns of neural response in the parents are shared with their offspring. Anyone interested in doing that, let me know. You've got a long climb ahead of you because the reviewers are not happy with that stance, but that's what needs to be done.
Great, thank you, Mary. We have a few more questions that have come in. So Dr. Ralph Wharton asks about the role of handedness or mixed dominance as a factor. I'm not certain which of those studies, but maybe you could speak more broadly about that, I guess. Yeah.
Yeah. Well, we didn't really have the handedness in the infants, but in
right.
the I mean, it's a great question though, because of course the dominance. I have to say that most of our participants are right-handed. In the child studies, we do include left-handers and mixed-handed. They're a small fraction of 10% of that, and we always try and control for that in our control groups. In the bipolar, the adult bipolar, they're predominantly right-handed again. We have historically excluded left-handed and mixed-handedness simply because of the problem with dominance. However, when we have looked at left-handers, when we have included left-handers, there doesn't seem to be I mean, there's not that many. That's the problem. We can't focus on just recruiting left-handers. So you can never do a sensitivity analysis which is truly sensitive because it's going to be predominantly majority right-handers. What I have to say, which is kind of, so I cannot say it's just it's there in all left-handers because we just haven't examined enough left-handers. What I can say is the right VLPFC just isn't showing this pattern at all in any of the people that we have studied. It's only in the left. And this, I mean, you could speculate why it's left. I mean, obviously it's the approach avoidance related story, particularly in right-handers, of course, not necessarily in mixed-handers. But as I say, we're not seeing huge differences. I'd love to have more left-handers in the study and the mixed, but we can't always find them. They're just not so common in society, or at least in the people who want to take part in the experiments.
Yes. I had one more question, and then I'll get to Richard Brockman's question about the implication of your findings for this DSM-5, new diagnosis in DSM-5 of disruptive mood regulation disorder, right? Which I think my understanding, I'm not a child psychiatrist, but my understanding was that it was added to avoid premature diagnosis of bipolar disorder in children based on more trait-like abnormalities, right? Trait like impulsivity, irritability, etc. Your findings are about, I think, some of those things, right? These behavioral traits that may predispose to the development of bipolar disorder. So I'm curious your thoughts about that diagnosis in light of these findings.
Well, it's kind of controversial diagnosis, wasn't it? And
yes.
I think many clinicians, many child psychiatrists, colleagues of mine who are full-time clinical child psychiatrists and psychologists as well, would think of it as a holding pattern. It's like, waiting at Heathrow Airport, waiting to take off. And you have it's like a holding diagnosis before, it's like a form fruste before something else emerges. You wouldn't necessarily diagnose an adult with it, so it's there. I think it's a fair point, because of course it was there to design to stop bipolar disorder being given as a diagnosis to younger children, and then all the medications, going back to the medication thing again early. But of course the downside is, of course, bipolar disorder might be missed. I think it's a fair point. I mean, I don't want to comment on the politics because it gets a bit messy. But I think you're right though. The features of it are similar. I won't say totally the same because this was really more. I mean, Alan Liebenluft obviously was one of the great proponents of this diagnosis, and it speaks more to the irritability aspect and frustration, which of course are there in people with mania risk, but it's not necessarily just focused on the sensation-seeking impulsivity per se, although it includes that. So I think it is what it is. It's a holding pattern. Some of those children are going to go on and get bipolar disorder. And yeah, I mean, you could argue that what we're doing, particularly in the children, is looking at the neural basis of this whole gamete, this whole cluster of externalizing like kind of symptoms that in turn could predispose. However, I would say, what we're really focusing on is trying to focus on, particularly in the adult stuff, is the more fine-tuned, predisposition to mania, the impulsivity, sensation seeking.
Oh, sorry, there's one question that just got moved. Let me find it. Oh, no. Oh, here it is. Okay, good. Our final question is from Richard Brockman, who says, thank you for an inspiring presentation. With a depressed caregiver and greater network default mode control of the amygdala, as opposed to the more active executive control of the amygdala in the infant, is the implication that the infant's default mode network is more inclined as she matures to allow the amygdala to regulate baseline mood and behavior?
Yeah, yes, kind of. I mean, I didn't have time to discuss that in the talk, but yes, what we say in the discussion, and what I thought long and hard about this. Yeah, I mean, if you think of it more as the influence of the task negative network, so undue influence on amygdala in the environment. So that would mean that internal world, internal thinking, ruminative style, all encoded by the default mode network, that kind of aspect of one's being kind of has an undue influence on the amygdala, and vice versa, because this is not directional connectivity. So that is a sort of proponent, as a sort of a prominent pattern. So rather than having a more of an executive control of the amygdala, where we have a top down, let's control ourselves and move on. It's more, let's pay attention to our inner world and inner thoughts. And of course, it doesn't, that you can see would not be an adaptive style, an adaptive pattern of response to cope with the trials and tribulations of the world, when sometimes you just got to switch off your inner world and focus and externalize. So in that sense, it could be very disastrous for you, because it prevents you from switching off your own internal world and switching onto the task now. So that's how we were conceptualizing, and of course showing that it's related to lower smiling in the infants kind of speaks to that. Similarly with the salience network, and the salience network about detection of what's important. If that's more in tune with the amygdala, it might mean that the amygdala is going to be more online with the salience network to sort of come in and perceive change, surprise, anything that's a change from the standard environment, and that might be a bad thing, as opposed to being able to regulate. I think you have to think about it as being a yin yang with the executive network. You've got more default network connectivity. If that, in combination with lower executive control network, that's going to be a bad thing, because the amygdala is going to have a free rein to be at one with the inner world and not be regulated, and so your focus of attention is going to be on yourself internally, with all the neuroticism that goes along with that, as opposed to externalizing and moving on.
Yes. And I'm sorry, one more question came in. I hope this is okay, from David Hellerstein, who asks, do many of these children have other circadian rhythm disorders, such as thermoregulation?
Interesting. We're not looking at that at the moment. However, what we these babies, and it's only a limit. There is a limit to what we can do. I mean, there's so much more we would love to have done. We assess them 3 times over 18 months, and we scan them twice. We're scanning them at 9 months too. So we don't measure that, but what we do measure is we have a questionnaire about their sleep, their circadian pattern. Now, arguably at 3 months, it's hard to actually gauge exactly what they're going to be like when they're children and adults, because a lot of babies spend a lot of time sleeping, but of course some do not, and that is particularly starts emerging in their first year. So we do have those measures, and we haven't looked at those in detail yet. But yeah, we have an adapted sleep questionnaire in Pittsburgh. We're interested in a lot of us are interested in sleep, so we've adapted one of the questionnaires for use for the care caregiver to assess their baby's circadian function and how that might in turn relate to this. I mean, obviously it's going to factor in, and that's another area of study, another examination down the line. But thank you for the question.
Wow, Mary, that was really just an amazing talk and discussion. So thank you so much.
Thank you. Thanks, Jeff.
All right, we'll stop there. Have a good day, everyone.
Thanks to everyone. Thanks for stopping by. Thank you.
Bye-bye.